Thanks for being here.
Hello there.
Hey, for Madisyn's talk. Madisyn is a senior in our department, the biology department. So Madisyn's research started off as a class project in my conservation biology class. I was really impressed with it. I asked her to go ahead. So she and the rest of her group joined a research project in my lab to go ahead with it. I'll let her talk more about her amazing project, but just some major unique points. There's very little research done on how plant species are being affected by climate change in the southeast, and so she fills that important gap with her work. And secondly, this is the first time that anyone's used these particular methods to approach this subject altogether. So let Madisyn take it away.
Hello everyone, my name is Barksdale, and today I will be discussing the effects of climate change on Alabama's native plant phenology. So species all across the world are responding to challenges posed by climate change in three broad ways. First, some species that are unable to cope with the warming and other impacts of climate change may go extinct. For example, the poster child of climate change, the polar bear. However, we do not have to wait for climate-driven extinctions. We already have examples such as this rodent native to Australia that went extinct in 2009 to 2014 due to habitat loss from rising sea levels. These species are fighting back by adapting, and for example, some are moving to places with more suitable temperatures. This includes moving higher in latitude towards the poles and away from the equator, or sometimes towards higher elevations. And some of these species are adapting by changing their phenology. Phenology refers to the different events in the life cycle of an organism. This could be the timing for breeding season, egg laying, molting, or even emergence from hibernation. In plants, we have spring phenology, when leaves start coming out. Fall phenology is when senescence occurs, and winter phenology is when they lose leaves. Studies have shown that with warming trends, spring phenology has advanced in a number of species, including our oak trees. As you can see from this graph, with advanced spring phenology of the oak tree, the winter moth has also matched the hatching of its eggs so it can feed on the young leaves. The caterpillars depend on these early leaves and have to hatch around the same time in large numbers to survive. Now, this may not seem like much of a negative impact as everything is still running smoothly in this system. However, this system involves another actor, a bird called the great tit that is dependent on mass hatching of these caterpillars to feed their young. As you can see from the graph, they have been unable to match the oak tree and caterpillars, and this has led to fewer offspring surviving. This demonstrates how changes in phenology may result in mismatches in essential species interactions in nature. Now, there are many ways that phenology has been studied, but one common way is using satellite imagery, like this study here that reported an advance in spring phenology by 0.9 days per decade. However, these studies are not able to capture species-level data and are completely absent for the ecologically vulnerable southeastern U.S. The U.S. is part of the NACP, which is one of 36 global biodiversity hotspots. Biodiversity hotspots are less than 2.5% of the Earth's land surface, and about 50% of all plants and animals. However, 1/3 of the species listed on the Endangered Species Act are from the Southeast, but we know very little of the impact of climate change on biodiversity in the Southeast. To address this gap, we asked two questions. First, have temperature patterns in Alabama's climate changed over time? And are flowering species showing changes in phenology in response to these climatic changes? To answer our first question, temperature data was obtained from NASA Langley Research Center. The data is based on GMAO and MERA2 assimilation products and include long-term average estimates of meteorological data. To answer our second question, phenology data was collected from vivarium collections across the state stored on the Alabama Plant Atlas. A vivarium is a dried plant specimen with collection details. Photos of each vivarium were carefully studied and collected notes on phenological data collected, including key phenophases. We did this for 11 species with primitive photographs, and additional metadata such as data collection, locality, scientific names, and web page links were also recorded. Data was analyzed in R&R Studio with the GT plot package. Temperature data was used to develop seasonal temperature data, defined as spring from March to May, summer from June to August, fall from September to November, and winter from December to February. For the phenology data, only flowering dates were evaluated, as it was hard to distinguish phenological phases between each species, and a Pearson's correlation test was conducted to test for the significance of the correlation between day of the year of flowering and the actual year itself. The results of our analysis showed that temperatures throughout the year and seasonal temperatures all showed an increase between 1981 and 2022. However, all categories showed a significant increase except for summer temperatures.
Just go ahead.
So all of the p-value can be seen on the screen and show the temperature data reported between each seasonal temperature. Okay. We compiled a data set comprising of 1,728 specimens for 11 species, spanning over 146 years. Among these, 1,126 specimens consisted of herbarium samples with flowers. Okay, this data set included 6 spring, 2 summer, and 3 fall flowering species. And some of our species had as little as 96 entries to as many as 264. And for phenology data, our y-axis represents the day of the year when herbaria was collected. So we have 365 days, and we only use herbaria with flowers, so this represents flowering dates. And the x-axis represents the year. And the p-values are the result. And so the downward trend line means that flowering seasons are advanced, and this was present in 10 out of 11 species, and mainly in 4 of these, so Seaver's Janica, and bivalve, and then R. hispidus, and Ilium canadense. The only one that showed a delay in the flowering season was T. subaspera. So to conclude, the results of our study showed an increasing trend in Alabama's temperatures, with significant increases in the annual average temperatures, and significant increases in seasonal temperatures except for summer, as well as an advance in flowering season for most species, and this includes 10 of the 11 species, with 4 species showing a statistically significant advance. Interestingly enough, all species showed a statistically significant advance were spring flowering species, with one species, T. subaspera, demonstrating a significant trend of delayed flowering. Our study is the first attempt to use herbaria specimens to investigate changes in plant phenology in response to climate change in the Southeast. We observed similar findings in numerous studies across the globe, and similar trends have been identified with satellite imagery. However, this lacks the capacity to capture species-specific responses, as some may be responding in different ways not seen using satellite imagery, which further proves the importance of our methods. And future studies would include a wider variety of species. And we would like to extend our thanks to the JSU Department of Biology and Dr. Pruitt for his advice on specimen selection.
Any questions for our speaker?
Hey, thought about mentioning the use of LiDAR and remote sensing technology, mainly more satellite-based tech and drones incorporated with LiDAR.
We didn't use that because we wanted to use species-specific level data. But I'm not sure.
specific sense. Yeah, so satellite data you can't really capture species level data, which is a set of their approach. You got data right at the species level. Any others? Any other questions?
So the hours that were in the summer expected, and they were in the same part of the other seasons.
It was mostly spring, flowering seasons that were affected.
I believe we have to look at like p-values, right?
Yeah, just for